Dear viewers, greetings. Today we can discuss about Codon Dictionary and its salient features. This table shows the genetic codes, totally 64 codons there. Here three positions. First positions are UCAG, as like second letter also second position UCAG, and also third letter also UCAG. In this way, three position alternately we can written as first position U means second position U, third position U, as like first two positions are U, third position are C, first two positions U, third position A, first two positions U, third position G. In this way, alternatively, first, second, third letters we can arrange it. We can get the totally 64 codons. Next one, genetic properties of codons. There are, the code is a triplet code. Second one is the code is non-overlapping code. Third one is the code is comma-less. Fourth one is the code is non-ambiguous. Fifth one is the code is polarity. Sixth one is the code is degenerative. Seventh one is some codes act as a start codon. Eighth one, some code act as a stop codon. Ninth one, the code is universal one. Now briefly we can discuss it. First one, the code is a triplet code. The code is a triplet code means the nucleotides of mRNA are arranged as a linear sequence of codons. Each codon consists of three successive nitrogenous bases. The code is a triplet codon. Next one is code is non-overlapping. The genetic code is non-overlapping. Non-overlapping means the adjacent codons do not overlap. Adjacent codon means nearest one. The non-overlapping code means that is same letter is not used for two different codons. For example, same nitrogenous, for example, A means that is only for code one codon, does not overlapping with adjacent one. In other way, no single base can take part in the formation of more than one codon. The picture shows the mRNA arranged as a nitrogenous bases, each three, which indicates the one amino acid, not overlapping with each other. Next one is codon is comma-less. There is no signal to indicate the end of one code and the beginning of the next. Here no comma or full stop there. Continually going the nitrogenous bases. The genetic code is comma-less one, comma-free one. For example, the mRNA arranged as CAT, GAT, in this way, continuously arrange it, no gaps there. Next one is the code is non-ambiguous. A particular codon will always code for same amino acids. Thus, one code cannot specify more than one amino acids. For example, AG means methionine, GUG means valine amino acids. One codon specifically indicates the only one amino acids. Next, the code is polarity. Polarity in the sense the genetic code has polarity, that is the code always read in a fixed direction. Fixed direction is which codes the 5 dash to 3 dash direction alone. Next, the code is degenerative. The code is degenerate, which means that the same amino acid is coded by more than one base triplet. Different codons which indicate the same amino acids. Example, the 3 amino acids, for example, arginine, alanine, leucine, each have 6 synonymous codons. 6 synonymous codon means different codons which indicates the same amino acids. For example, arginine means 6 different codons indicate arginine, as like alanine and also leucine. Some codes as act start code. Translation always begins at a start codon. During protein synthesis, the AUG initiation codon, which corresponds to methionine, this only the initiate the protein synthesis process, which has sequence AUG and encodes the amino acid methionine in some organism, most of the organism. Thus, every polypeptide typically starts with the methionine, although the initial methionine may be snapped off in later processing steps. Alternative start codon depending on the organisms include GUG or UUG. These codons normally represent valine and also leucine respectively. But start codons, they are translated as methionine or formyl methionine. Methionine for eukaryotes, formyl methionine for prokaryotes. Some codes act as stop codon. The 3-letter codon, stop codon, namely UAG is a amber, UGA is apple, sometimes also called as amber, and one more thing, UAA is occur. These are the stop codon, are called as termination or nonsense codon, while the 61 codons are sense codon. The codon is universal one, although the code is based on work conducted on the bacterium, especially Escherichia coli, but it is valid for other organisms also. This important characteristics of genetic code is called as universality, universal one. It means that the same sequence of 3 base encodes the same amino acids in the all life form of simple microorganism to complexed one, multi-celled organisms such as human beings also. These conditions, some exceptions there. The exceptions are the genetic code once thought to be identical in all form of life has been found in diverge slightly in certain organisms and in the mitochondria of some eukaryotes alone. For example, in mitochondria, AUA is the codon for methionine instead of leucine. This is in mitochondria of eukaryotes. UGA is the codon for tryptophan instead of being the stop signal. AGA and also AGG are stop signal instead of codon for arginine. This is in mitochondria. Next one, degeneracy of genetic code. An amino acids can be coded by more than one codons. This is called as degeneracy of genetic code. The table shows the totally 64 codons. Here 21 codons indicates the 20 amino acids. Next one, verbal hypothesis. Crick postulated the verbal hypothesis to account for the degeneracy of genetic code. According to this hypothesis, the first two bases of codon pairs according to normal base pairing rules, with the last one base of anticodon base pairing at the third position of codon is verbal one. The picture shows the first diagram. mRNA consists of CUC, tRNA consists of GAG. This is for normal pairing one. Instead of this, verbal pairing means mRNA consists of CUU, tRNA consists of GAG. This is for third base pairs are verbal one, not complementary to each other. In this way, pairing means this is called as verbal hypothesis. Next one, verbal hypothesis explains the degeneracy. Verbal hypothesis explains the degeneracy of genetic code, that is, existence of multiple codons for single amino acids. Although there are 61 codons for amino acids, the number of tRNA is far less, around 40 only. So that the verbal hypothesis or verbal degeneracy will be taking place, which is due to verbling. Next one, biological significance of degeneracy of genetic code. If the code were not degenerate, 20 codons would designated amino acids and 44 would lead to chain termination. The probability of mutation to chain termination would therefore be much higher with a non-degenerate code. This picture explains the total flow of genetic information. Generally, DNA to DNA replication. After DNA synthesis, the RNA is synthesized by transcription. After synthesis of RNA, which help of translation process, which produce the proteins. This is what genetic information carry out from the DNA to protein. Next, clinical significance. Mutation can be well explained using the genetic code. Here, two types of mutation. First one is point mutation. This further classified as silent, missense and also nonsense. Second category, frame shift mutations. First one, silent or silence mutation. Single nucleotide change. For example, A to G, same amino acid is incorporated, the mutations going unnoticed. For example, silent mutation means ATG, GAA, GCA, CGT. Here second amino acid, glutamic acid, it will be the GAA, the third nucleotide changed as a GAG, which is also indicated the same amino acid. So, this is called as silent mutation. Next one, missense mutation. The single nucleotide change, for example, A to C, different amino acid incorporated. Loss of functional capacity of protein. Here, single nucleotide changes also indicates the Different amino acid means which lost the function of protein. Next one, nonsense mutation. The single nucleotide change from C to T, which forms the stop codon, is generated. In mRNA represented by UAG, premature termination of chain may be incompatible with life. During the change of individual nucleotide bases, which indicate the termination codon. This is called as nonsense mutation. Final one, frame shift mutation. Here either insertion or removal of bases can alter the reading frame with the resultant incorporation of different amino acids. The picture shows the methionine, glutamic acid, alanine, glycine. Here second one, glutamic acid consists of GAA. Here GAA, the G will be removed, means the reading frame shift will be shifted. Instead of GAE, it will be translated as AAG and CAG, then GT. In this way, frame will be shifted, means the amino acids, different amino acids will be indicated. Dear viewers, thank you for your support.